essays. I do.
From Microbe TV, this is Q&A with A&V. I'm Vincent Racaniello, and joining me tonight from New York, Amy Rosenfeld. Hello again, Amy.
Hello, Vincent. How are you today?
I'm green and you are purple. It's a good combo. It is actually the part of the color combination at the incubator.
Yeah, but I'm also green and purple.
Oh, look at that. We have green underneath.
Yes,
Excellent.
we always have multiple layers.
Thanks everyone for coming tonight on an off night. Amy was traveling yesterday, and so we appreciate your coming. Let me thank the mods before we start. Les is here tonight, and I saw Tom as well, and I believe I saw Steph as well. And Vanity says that she's going to be up in the air, so she'll be on and off. So thank you all. There's Steph. Thank you all for coming tonight and having a nice coffee clutch about viruses. Is that what you would call it, Amy?
Coffee clutch? Like
Yes.
a clutch?
So here, I want to remind you at the outset, please support Amy's work on enteroviruses, and you can do it in lieu of supporting Microbe TV. That's fine with us. And you go to giving.columbia.edu, you click Make a Gift, which is a little bit down from the top of the page. You type enterovirus, and the right link will come up, enterovirus D68 research, I believe. And you click it, and then you can enter your gifting information. So Amy would be grateful to have your support for work that is very important, unusual, unexpected, and needs to go forward to keep us all safe.
Yeah, it does. I mean, so, most of my conversations over the last couple of days has been the fact that eradication is not going all that well.
Eradication of polio?
Yeah, not going all that well. And the fact that it was anticipated that childhood paralysis would go away, and it surged instead. So,
you think it has anything to do with COVID?
No.
No?
No.
Okay.
What do you mean? What has to do with the COVID? Surge of AFM? No, childhood paralysis surged way before COVID.
What about the
It
recent
continues.
polio cases? Do you think they have anything to do with polio and not being able to immunize people?
Nope.
Yep. Okay. All right, here we go. Yes.
They're trafficked from, where was the Israeli case from? I forgot. But the Malawi case was because a visitor came from Pakistan. And
correct.
It's really hard to, the eradication campaign in Pakistan has not been so successful. It's really hard to do it, just like it was really hard to eradicate polio in India because of the temperature and the climate or something, I believe is the current thought, right?
Also, in India, they required many doses to get some immunity, and they got it. And maybe in Pakistan and Afghanistan, part of the issue is combat is preventing people from immunizing. So there's a lot of wild polio type 1 circulating there.
Yeah, but that doesn't have anything to do with COVID. They've been under warfare, tribal warfare for 4,000 years.
All right, Jamie says, new study out of UK shows even asymptomatic COVID can cause brain shrinkage. I was warned when the pandemic started about brain damage from COVID. Is it common for a virus to damage brain, Amy?
I saw the paper. I skimmed it. Yeah, it's kind of not so good data. Not really. Yeah.
Daniel talked about it a bit tonight in his update.
Yeah.
The good part about it is they had a series of patients who had a brain scan, and then they got COVID, and so they gave them a second brain scan. So they had a baseline, which they could then see what happens after COVID. And it doesn't look like. I don't know what the functional consequences of the scan observations are. Okay, we just don't know. But yeah, it looks like in mild to severe, you get brain alterations. But I wonder if that happens with a lot of virus infections because of cytokines and inflammation and so forth. We just don't know. I don't think we've ever done anything like this before.
I don't know. Some group in the Netherlands wrote a review in Trends of, I think, immunology, neurovirulence, neuroinvasion, and neurotropism of SARS-CoV-2.
Mm-hmm.
I'm not.
It doesn't neuroinvade, does it?
So a lot of their conclusions were, yeah, it's like neuroinvaded because you can find a protein stain or an RNA stain or something or other. And if you looked at it, so it's kind of like the argument that Ian had about the antibodies for EV68 in the CSF, right? You don't know that they're really made there or that they were recruited there, right? You'd have no idea that they're intrathecal. So I mean, so what if you have a mild antigen stain? I mean, it's not infectious virus, right?
Well, that depends on your definition of neuroinvasion. If you feel that infectious virus has to enter, then that's hard to prove, right?
Well, then everything's neuroinvasive because everything eventually can just
T cells are neuroinvasive. But in kids with mumps in the day before vaccines, 50% of the kids had mumps RNA in the brain.
Yeah, I know. I talked to somebody about that. So we talked about this yesterday.
And apparently, well, I don't know if it was infectious virus, right? You just don't know. But it certainly didn't cause any problems. And if we had done scans, maybe their brains would have swelled. Who knows? But I think the jury is still out, as Amy says, on this observation.
Yeah, and so like they also said, like, because a few papers can show that there's RNA in, like, some autopsies that we looked at. We did those papers on TWIB. They're like, oh, clearly it's neurotropic. It has potential for being neurotropic. And then they talked about neurovirulence and like using iPS cells and stuff, and some other models. And many things can infect a cell when it's in a petri dish. Does not necessarily mean that is true proof of what is occurring in if the biology doesn't support it.
If you remember, we had Kieran Thacker on, and she and Jim Goldman authored a paper where they did a lot of autopsies and concluded there's all the damage is hypoxia or inflammatory based. There's no evidence for virus reproducing in the brain. Do you remember?
Yeah, I remember because it followed Carlos's paper, and she was trained by Carlos. And Carlos also came to the same conclusion. It wasn't neurotropic or virulent or invasive. So remind me, in some mouse model of SARS-1, the virus, ACE2 transgenic SARS-1 goes in the brain, but that's thought to be aberrant, right? But I don't know. What is it, the K12? Is that the transgenic or is that the adenovirus?
I don't remember. It was the one that was made.
It's by Perlman.
Perlman, yes, right? It was Perlman, right? And he says in his paper this is not
an
a
artifact.
Human, and an artifact, yes, of the mouse model.
Yeah, it's not it doesn't recapitulate human disease.
Double vaccinated April, hesitant to get the booster due to inflammation of my sciatica after dose 2. Do I have enough protection with 2 doses of Pfizer? What do you think, Amy?
So if the 2 doses were following the very short interval, no. I mean, she's protected, but she'd be more confident if she got the booster. And it appears that the side effects of getting the booster are not anywhere near as painful or detrimental as the side effects of the second dose to begin with.
Yeah, that's correct. That's absolutely right.
So if she was the 3 weeks away, blah blah blah, then yeah.
Jamie writes, I know a brain injury lawyer who does huge multi-million-dollar cases. His experts warned him when pandemic started that brain injury was likely. Why was this not public knowledge? Because no one knew brain injury was likely. And if whoever said it didn't know what they were talking about. And they're not experts. That's what I would say. What do you think, Amy?
His experts? Who is his experts? The people with the crystal balls that have the fog? When the lady was the witch and she would watch Dorothy and she would have like the crystal ball in the middle of her cauldron and stuff? Interesting.
Yeah. You have to be very careful about people who call themselves experts. Oh, by the way, I have to tell everyone this story. I think I already told Amy. Yeah, I sent you the email. So last week, someone wrote, such and such a person at Yale says the only way to control COVID is by sterilizing vaccines and masking. And they said, what do you think? I said, well, that's nonsense. It turns out the guy was watching. And he emailed me and he said, I never said that. So when you guys tell us...
I was thinking that you were going to talk about how Lex told you nobody should be an expert.
No, I wasn't going to talk about that.
Oh.
So basically, I have to be careful when you folks say, this person so-and-so said this. I can't verify it here on the fly. So I have to be careful and say, okay, if that person said it, then I don't agree with it. All right, that's how we have to put it. But you could say at the beginning of the outbreak that the virus would do anything. And now there's some long COVID. You could say you were right, but that doesn't mean you knew anything.
What are you talking about? You could still today say that the virus does anything. Anything you want it to say.
Yes, of course. It's a big problem. It's a real
problem.
I
know.
Oh, someone lost their father to long COVID. I'm very sorry about that.
My condolences.
When influenza A packages its genome, does it usually package the 8 correct segments? So, Tess, there are 2 thoughts on this. So if you just randomly grabbed 8 segments from an infected cell, 1 in 400 particles would get the right segments. And that's about what the infectivity ratio is for influenza. However, it's probably not random. There are packaging sequences at the ends of each RNA which direct them into the capsid. So that's how it works. With many viruses, there are sequences on the genome that interact with structural proteins that direct the genome into the virus particle. There's quite a big literature on that for influenza virus.
Yep.
People are very sad.
Why?
Oh, about someone's father died of long COVID who comes here a lot.
Oh, yeah, that's not so good.
FYI, Bay Area dwellers, there's a paramyxovirus outbreak in San Francisco at the moment among wildlife. Protect your pets. Distemper, indeed.
Not so good.
No, not good.
Get your dogs vaccinated.
Did you get my email with links to questions about bovine leukemia virus connection to multiple cancers? I did. I am sorry I didn't read it today. It was just a long list of stuff, but I will take a look. If you come back next week, I will let you know our thoughts.
Yep.
And tonight's a good night to have your question answered. There are just 400 people here, which is good. You can have all the questions answered.
All the questions. Let's not get carried away here.
Many. Purple sweater. Andrew guessed right, but it's not a sweater, is it?
It's a cashmere something.
It's cashmere?
Yes, it's a cashmere blend for something to go skiing. I bought it when I lived in Montreal.
How long does immunity last after 2 shots in infection? Amy
For
sent me a paper.
as long as you want it to.
You're going to get boosted. You're going to get infected, and you're not even going to know it. But Amy sent me a paper last week, which almost made the cut for tomorrow. It said basically, infection before or after vaccination gives you great immunity, the ability to neutralize many variants. So she had 2 shots in infection. I think that's very good. How long it will last, I don't know. The mucosal viruses, the memory doesn't last forever, but it will certainly last for a number of years. But I do think you're going to be boosted periodically, and you don't have to worry about it. Does the decrease in the incubation period of successive variants promote immune evasion?
I'm not clear that there's a decrease in incubation period.
Yeah, I don't know where that's coming from. I don't know where that's coming from. No. Immune evasion is promoted by changes in the spike that make the antibodies don't neutralize very well. Vincent and Amy, thoughts on colleges in New York lifting their mask mandates? Well, our college hasn't. Has Columbia lifted our mask?
Of course. It lifts on Monday.
Really?
Do you not? This is why I get annoyed, is because I send you the information and you don't read it.
When did you send me this information?
I texted you this information last week.
Columbia University is lifting its mask mandate?
Yeah, on Monday, March 14th.
Oh, so I don't have to wear a mask in class anymore?
No, I would wear a paper bag, though, tomorrow.
Why is that?
Because I'm going to yell at you. Because there's no point in me sending you stuff to inform you and educate you if you are going to continue to ignore it and be ignorant.
Well, that's good news. I think it's fine in New York City. Yes, I do think so. I think, as we've said before, we're vaccinated. The infection rates are low. Yeah, it's good. But what about New Jersey Transit and the subway, Amy? It's still a federal mandate?
I don't know anything about New Jersey Transit. The subway, yeah, you still have the mask.
Okay. All right. With respect to human papillomavirus sterilizing immunity, if antibodies contract everywhere else, why would you expect mucosal to remain high enough to always remain sterilizing?
Well, it doesn't always remain sterilizing. It was a mistake to say that it remains sterilizing, because it's not.
Who said it is not sterilizing, or what's the evidence?
FDA.
Oh. Because some papers say it's sterilizing.
Well, according to the FDA and the information that the company has sent them, no.
I see. So you can be infected.
Yes.
Okay. All right, there we have it. So there are no sterilizing human vaccines, Amy, correct?
Right.
What role does hemagglutinin and neuraminidase play in flu virus infections? Are red blood cells competitive in binding to HA-reducing viral load? No. The virus remains mainly in the respiratory mucosa, so it's not in the blood, and so red blood cells don't have any impact on reducing viral loads. Hemagglutinin attaches to the cell receptor, and when the endosome pH goes down, it catalyzes fusion of the viral and endosome membranes. So that's what the HA does. Very important. And the neuraminidase removes sialic acids from the newly made virus particles so they can leave the cell and they don't stick to the receptors on the cell surface when they're made. Those are good questions. I like that. And someone asked me in class, Amy, yesterday. Why does Amy think it's going to be another 5 years? Is that right? The pandemic. And here we have a question about that. Total 5 years.
Total
Yeah.
of 5 years.
Yeah. So regarding the prediction for another 4, not another 4, but a total, what will cause it to end? What changes between now and then?
The global situation. So the American public has decided that at this year, what is it, year 3, that the pandemic is over for the American public. So therefore, it should be over for the world. But that's not really true.
Actually, tomorrow, March 12th, 2 years ago, the WHO declared the pandemic. And we're in the 3rd year, yes.
Yeah, well, so that's not really true. And if you look at the history of the great pandemic of 1918, we did the exact same thing. The American public decided after 2 years that it was done with the pandemic, but there were still many waves, and some of them were greater than any of the initial waves for several years later.
Correct. I think that it's mainly immunization of many countries that don't have immunization so far that's going to end it.
Well, the Americans aren't going to wait for that. So
no, we will say it's over here, but WHO won't until the world has it over. Paul Offit had an op-ed in the Philadelphia Inquirer. Yes, and in fact, his wife, Bonnie Offit, emailed it to me.
Oh, and what did it say?
Not to me, to TWIV. And she said, as you know, I'm a fan of TWIV.
Well, what did that? I don't care what she wrote to you. I want to know what the op-ed said.
Get vaccinated. That's how this pandemic ends, and especially kids. Kids, because there's some states, as you know, that say we shouldn't vaccinate kids. At least
would that be another text I sent you?
That would be Amy this morning.
Yeah. Because the Surgeon General or whatever he is of Florida came out and is contradicting everybody else. And this is in line with his governor. And yet they've had, the most deaths ever or in total of the states. So it's a good plan.
It's ridiculous. We heard that he yelled at some high school kids, the governor, to take off their masks at his press conference.
Yeah, it was at the end of the article. Yeah, he yelled at them and said that the masks don't do anything.
He said they were hygiene theater. COVID technical lead Maria von Kerkhove informed that WHO is aware of a recombinant in Europe combination of Delta and Omicron. Thoughts? What do you think, Amy?
Recombinants occur all the time.
Yeah, we just picked this one up, right? That's basically it. So I'm not surprised. Neither is Amy.
No, I'm blinking because I have to make sure, I have.
Why are you blinking?
Just because, I just don't get the hysteria.
Tyler's University, fully vaxxed and boosted students and staff is making us mask, but nowhere else. In classrooms, but nowhere else.
Maybe because there's no circulation of air and the door is closed and you're right on top of each other.
Yeah. Environmental says, my Wednesday morning coffee shows on climate and environment are called Coffee Klatch. Yeah, this is Cafe Klatch is the original name, I believe. Cafe Klatch. Do any of you have cats? I don't. Amy, you used to have a cat, right? Or you had a dog? No, right? When you were a kid. My question
is... The
dog
was mine. My sisters had cats. The dog was mine. Carnegie was mine.
That's a great name for a dog, Carnegie.
Yes, he was adorable. He was dressed in a tuxedo because he was a German Shepherd Beagle mix. And he looked like he was always wearing a tuxedo.
Why did you call him Carnegie?
I let my sister name him, and that's what Wendy came up with.
Yeah. Anyway, question is, can they transfer lots of germs and viruses if they go on beds and tables after they use the litter box? everybody has germs and viruses that we transfer amongst each other. In fact, the cats have a microbiome, and people who have cats, their microbiome tends to trend towards the cat microbiome. You get their bacteria, and I'm sure you get their viruses, most of which have no effect on you. So let them get on the bed, no problem. If you try and keep them off, that's the real thing. Unless you put a fence up. Hey, Neva, how are you doing? Haven't seen you in a while. Neva from Buda. Good to see you. Thanks for visiting. On Slavin's podcast, David Ho expressed concern about SARS-CoV-2 escaping Paxlovid if prescribed too much. Gee, I wonder who said that months ago. Not on Slavin.
Slavit.
Slavit. Andy Slavit.
Yeah, it's S-A-L-A. It's S-L-A-V-I-T-T, I believe.
Yeah, I think that resistance to a protease inhibitor is a reality, because that's what happened with HIV protease inhibitors. So you have to be careful. And
Isn't that what happened with Hep C?
Hep C as well. So, yeah, people are taking it left and right. Although, yeah, I'm concerned also, and Amy and I expressed it months ago. But we're not on Slavit, so that's fine.
That's because we didn't get to go to the NBA All-Star game in Cleveland.
Who did that? David Ho?
Yeah.
Oh, I could care less.
So Ho and Slavit sat next to each other at the NBA All-Star game in Cleveland.
Good for them. Carlin wants to ask a non-COVID question. Absolutely, of course. You don't have to ask our permission. Yeah, so I'll look for it. Let's see if it comes up. Are there any viruses that cause hair loss? An interesting question. What an interesting question. So, as you know, and you can answer this too, Amy, stress can cause hair loss. So if you get stressed
Really? Would that be in relationship to the series of 11 photographs that I've sent you and Jonathan over the last 9 months of my ever-growing bald spots? Would it?
Yes. And in fact, your hair loss due to stress was the first time I'd heard that was a thing, okay? And I saw the evidence. So if you get a virus infection that stresses you, and they all do, they cause cellular stress, then many could cause hair loss. Yes. I bet if you lost your sense of taste and smell and you didn't get it back, that would cause you to lose your hair. So, I don't know any that would replicate specifically in hair follicles and cause hair loss, but there could be global effects. Right, Amy?
You think? You think? Well, when was the last time somebody told you that your hair follicles were depressed? Like they told me mine were.
Someone told you your hair follicles were depressed?
Yeah.
Oh my gosh. Do you have to take
They suggested that I use CBD shampoo or oil, and I was like, no.
Why? Okay, never mind. Jeez. Do you think masks had a negative impact on our immune system since they limit our exposure to pathogens? I don't think so, because they're not 100% blocking anything. And then you don't wear them 24/7. You go home, you take them off, and you get your exposure to pathogens. What do you think, Amy?
Yeah, I'm not clear that it had a negative effect on our immune response.
It certainly had a negative effect on influenza transmission, though, right? That was great.
Yeah, for sure.
Robert is participating in a respiratory syncytial virus vaccine trial. I'm certain I got the vaccine due to how I felt the day after. Is there any way of making the placebo generate an immune response?
Sure. That's what the placebo effect is.
Well, sometimes they give you just adjuvant, depending on what's in the real vaccine. If they have an adjuvant, if you're really smart, you make the placebo with just adjuvant, and you will get inflammation and feel the same. But not many trials do that. I did see one and I was very impressed that they did that. Wouldn't you agree that that would be the way to do it, Amy? So you couldn't tell?
If the vaccine has an adjuvant,
Yeah, if
not
it
all
didn't, of
vaccines
course.
have an
Yeah.
adjuvant.
Yeah. Hello, Animal Party. No worries about being late. It's okay. Welcome back to the party.
Yeah.
So Animal Party, the handle, I think, was her kids.
Yeah, it's her 10-year-old son. And then she took it and he said, Mom. And so then he got a new one to make videos on YouTube. Yeah. But
Why didn't she make her own?
we love Jen. Jen is great.
Do pet dogs get significant symptomatic COVID and any documented transmission from dogs to humans? What's the story on dogs, Amy?
I don't believe that they develop symptoms and I'm unaware of dog to human.
Yeah. I'm not aware of it either, so not clear that it's an issue. But cats can get symptomatic, is that correct? Because the big cats in the zoos get symptomatic COVID, right?
Yeah, I think so.
Do you know anything about recent release of documents, FOI, as problems with the Pfizer vaccine? If so, are you concerned about the results? I heard of this a few months ago, right, Amy?
Okay, I don't remember.
You didn't hear about it, and I
didn't read them. If I heard about them, I didn't read them, and I'm not really concerned because if there were problems, the FDA and everybody would have retracted. The vaccine would have been withdrawn.
Yeah, there are no problems. I mean, there may have been problems in manufacturing. Oh, this batch didn't work today. We have to modify this process. Yeah, but if that's what you're saying, that could be interpreted as a problem, but it's a normal part of the development process. So, as Amy said, they're still being used. They're fine. Is there any implication for virus persisting in testes in men?
What virus?
Any virus.
Zika.
Zika can cause sterility. Is that correct?
I don't know if it causes sterility. Well, yeah, I guess it does because it damages the sperm. Yeah.
So that would
And
be
isn't
one.
Ebola also persists in the testes?
Yes. And the reason is it's an immunoprivileged site, so there's not a great immune response, and so viruses can go there. It's also the aqueous humor of the eye. The inside is also immunoprivileged, and that's how Ebola virus hangs out there as well. But yeah, in the testes, the worst outcome I think would be sterility. No, maybe that's not the worst. You've probably answered this, but is a third booster in October still going to provide protection for an autoimmune plagued person?
Depends on what the autoimmune disease is. It's not. There's tons of autoimmune diseases, so you have to make sure you know what your disease is.
Yeah. New York City restaurants are not checking vaccine cards anymore. It will be harder to get a good table at your favorite dining spots. Well,
yes,
everything is getting more crowded. Yeah.
Yes.
But I don't dine out, so it's not an issue.
You will.
The trains are getting more crowded. That's for sure. Totally.
Well, last night several people were not masked on the train.
Really? Did the conductors do anything about it?
No.
Because this morning the lady was yelling at everyone. She said, you have to have your mask on. If you don't, at the next stop, you're off the train. It was great.
Nope.
Know anything about Deltacron? Is there a recombinant between Delta and Omicron? I think the person just asked us that.
That's what the person just asked us like several minutes ago.
But the significance is not known. We don't know of any unusual properties associated with it. It's just there. Okay, that's all we know at this point.
There's lots of things that are just there.
Yes. And if you look, you will discover, right? Like polio in the sewage in the U.S. is probably there, but we don't look for it.
Yes.
For TWIV, you should interview Tim Spector and the Zoe Symptom Citizen Science app. It's a great example of big data with hundreds of millions of data points. I've never heard, but I will look up Professor Spector. Have you heard of Professor Spector, Amy?
No, but I'm not clear what he's going to tell me.
What about Mpro cleaving Nemo?
Okay.
What about it?
So it turns off the innate immune response. Okay. Sure.
Yeah, it's an antagonist, right?
Yeah, sure.
When I was in Florida last week, it was as if there was never a pandemic. My mom said a lot of her vaxxed old lady friends already got it, including her back in October. Okay. That's another world, Florida. Rainey received oral polio vaccine in the U.S. in the early 60s. Have had weakness in one leg for years. How do I determine if it's caused by the vaccine? Well, did you get the weakness after you got the vaccine? Was it within a few months of getting the vaccine or a few weeks? Do you remember? Or was it there before? Did it happen a year later, 2 years later? You may not remember, but there's no test you can have to determine that. But if you recall, if it happened after the vaccine, it might be a vaccine-induced paralysis. You must have been young then, right? I would think, Amy, that if a child got vaccine-induced paralysis, they would be all on it, right?
I would have thought so. I learned that we know somebody who got like 60 doses of OPV.
Why would they get so many?
Because every year their mother gave them a dose at the beginning of flu season.
Oh, I know who that was. Wow, that's a lot of OPV. Holy cow.
Yes, it is. And he's very tall. He's very tall and lanky.
And healthy.
Yes.
Oh my gosh, 60 doses. Holy
yes.
cow. I am finishing out a mostly asymptomatic infection. 3 Pfizer doses, high risk. I'm a believer. I wasn't offered Paxlovid. Should I have been?
No.
No, but unless you had comorbidities that they were worried about. If you were immunosuppressed or if you had, what's the biggest one? Diabetes?
Diabetes and obesity.
I missed the beginning. Did I miss a notification about moving to Thursdays? No, it was just for tonight because Amy was traveling, Doreen. It will be next week, Wednesday. And we will do it at 8 next week because I don't have class. Next week is spring break. Is that okay with you, Amy, to do it at Wednesday at 8 next week?
Sure. Well, so then Thursday morning you have to go to Ian's.
It's fine. I have to? Why?
Well, first of all, he requested seeing you. Second of all, he's the one who's talking.
Okay, I'll go.
And third of all, they don't like it when I ask too many questions.
So you're not going to ask questions next week?
No, I will. But, I ask questions that are difficult. Like, so they talked about tick bacteria and stuff, and somebody said something, and I then asked if it was vertically transmitted, and the speaker looked at me like, vertically transmitted? I was like, yeah, vertically transmitted. And then I wanted to know if they had thought to have a tick colony, because just finding RNA in the tick for the 7 different tick-borne pathogens that infect humans is not sufficient. You need to say that there's the infectious agent there. And they looked at me and they were like, why would I do that? And I was like,
so what Amy is
because
there's a lab meeting group of Ian's group, and he invited us to go. And it's Thursday at 10 a.m., which is a difficult hour for me to get there, but it can be done. So I will go. But the problem is I have office hours at 11. So next week I don't.
It's spring break, so it doesn't matter.
Yeah, I have to go. I'm
in a timeout. What does this mean? I'm in a timeout.
I don't know
why.
Tom
Hall said he's in a timeout. I don't understand what a timeout is.
Well, the moderators give you a 300-second timeout if you say something that they don't like.
Oh, okay.
Okay, Ian wants to know if the protein adjuvanted Novavax would be preferred for young adult males. Oh, to get around the myocarditis. Perhaps?
What myocarditis has been associated with Novavax?
No, with mRNA vaccines as an alternative. Right? Because if you
Also,
didn't...
then what is the protein adjuvant in Novavax? I don't understand the question. I'm very confused.
The Novavax vaccine, the protein-based Novavax vaccine, would that be an alternative? Right. Because it's not going to cause myocarditis. But we don't know until it gets into millions of people, right? Because that side effect was so low, we didn't pick it up initially in the phase 1, 2, and 3. So it could be. That's what I would say. Does COVID cause nerve damage or brain damage? Well, it can certainly cause neuropathy, nerve pain, right? This study, the imaging study out of the UK, was it? Suggests that there's some alterations in the imaging of the brain in COVID patients. So maybe yes. Flu is rampant here. COVID non-existent. Where are you? German opera singer? Assume you're in Germany. Do respiratory virus outbreaks take turns? I know they shouldn't compete. Can recent COVID infection activate innate immunity to make flu infection less likely? Amy is the expert on this. Can you have multiple respiratories?
Yeah, you can have multiple respiratories. And COVID-19 does not, SARS-CoV-2 does not stimulate the innate immune response, according to Ben Tenuver and various others.
Okay, so here's Carlin's. I had dengue fever, spent a week in the hospital. Even though it was not fun, I made it. I've always heard you can catch dengue a second time. That's correct. And here's your continuation. And you might not survive it. So my question, is that true? Yes. So you can die of second dengue infections if it's, yes. And should I take it Dengvaxia? Ah. So Dengvaxia is not advised if you've never had dengue. So kids less than 5, you shouldn't take it. Because it will then act, since it's an infectious vaccine, it would act like the first dengue infection. And therefore your second one could be bad. But you have had dengue already, so you could take Dengvaxia. If you want to, if you live somewhere where there is dengue and you're at risk for getting it. But if you live in Minnesota, no, you don't have to.
All right, it's 8:43, so like 2 more questions because I have to do plaque assays to put overlay on.
So Kathleen emailed me about unexplained gastro issues. Oh, I think I forwarded that to Daniel. And hopefully he will get back to you, Kathleen. Let me know if he does or doesn't. You can send me another email and I'll bug him. So Doreen says, I know it will be time to move on to other subjects eventually, but I'm not near tired of seeing A&V. Oh, that means she's going to be tired of us at some point, Amy. But we'll always have viruses to talk about. And I'll bet even after COVID, there'll be 100 people here. Maybe we'll make it less frequent, but there'll be 100 people here because it's a community. As long as Amy can do it. We don't know if she's going to be able to continue, right?
Right.
Okay, here's a question. I'm a woman between 30 and 40, high blood pressure, 2 shots of Pfizer. I'm offered a booster of Sputnik. Is the risk of blood clotting worth it? Sputnik is not associated with blood clots, is it? That's not an adenovac. Oh, it is. It's an adenovectored vaccine. Sorry. Yes, it is. I don't think you get the blood clots on the subsequent. Well, I guess that would be the first boost of the Sputnik, of the adenovectored. So you have 2 shots. You have reasonable protection. I don't think you need to have Sputnik booster. No. What do you think, Amy?
I'm not a big fan of boostering, so.
All right, 9 pages of 1,300 adverse events released from Pfizer. Anything to worry about?
9 pages of 1,000. So They're saying that there's 11,700 adverse events reported out of
1,300 over 9 pages.
right. So that over no, there are 9 pages of 1,300. So that means there is 11,700 adverse events reported because every page has 1,300 adverse events listed
Okay,
on
well,
it.
I don't know how you get so many on one page, but no, I don't
I
think
don't know.
it's
I don't know.
anything to worry about.
I'm not clear, but so that's 11,000 out of several hundred million doses, right?
Yes. I don't know what these are, whether these
So
are.
let's just, it doesn't matter what they are. Just do the math.
It does matter
So
because
1% of 100 million is 1 million. And 1% of 1 million is 10,000. So they're at like 0.1 times, so they're at like 0.001%, which probably ranks with how fast you're going to get run over by the bus walking down the street.
the other problem is if these are reports from the Vaccine Adverse Events Reporting System, VAERS, then they're not verified. They're just what people write in. You could write in anything you want. But as Amy says, even then, the frequency is very low. So we're not
It's
concerned.
probably it's probably on the same level of, getting
Yeah.
hit by the bus.
No, where is this? Amy, you became a lot cooler to me when you said one of your favorite bands was Joy Division.
I didn't realize I was not cool to begin with.
No, he said a lot cooler.
Okay. Yes.
What's the outlook for regions or countries that have not yet had significant outbreaks and or vaccination programs?
All you have to do is look at Hong Kong.
Outbreaks.
Well, they didn't have significant outbreaks because they had a zero tolerance policy, and the old people refused to get vaccinated, and their vaccination program sucks. So they have the highest deaths right now and cases. So, I mean, it doesn't take a rocket scientist.
A wants to know how your interview went.
Well, we can all sit on pins and needles like I am and wait until they tell you.
Yeah, guess it went well, but she's waiting.
Okay, 2 more questions and then I have to go.
Is it fair to say the pandemic is over for the vaccinated?
Sure.
Another Pfizer documents. We don't need to address that. At what point of new cases per 100,000 are masks and packed churches no longer necessary? Many are anti-vaxxers.
Doesn't matter if they're anti-vaxxers. If you have less than 1 case per 100,000, no mask is required. When you get to that number, call me.
Purple is the royal color. Amy is the queen of virology then?
I don't know if I'm the queen of virology. I'm just a virologist.
Yeah. Amy, do you think the recombination level people are now verifying in SARS-CoV-2 can help explain far-flung variants like Omicron?
Far-flung variants? Well, those mutations had to arise in the genome somewhere, right?
Yes. I mean, certainly there's a contribution of recombinants, right? But
There's certainly a contribution to recombination events, yes, but they had to be present in some genome at some point without
yeah.
a recombination event.
Yep. Have there been any papers showing the 30% more transmissibility of BA2? No. No. Do you know the original Omicron? They said no intrinsic transmissibility increase. It evades immunity. That's why it has a higher secondary attack rate. Would you agree, Amy?
Yeah. Not clear that anything has a higher transmissibility rate. All right.
Now
Last one,
now
and
they're
then we have
calling
to
me
go.
now they're calling me paper bag. It's okay. I have a good sense of humor. Don't you think, Amy? Don't
I don't
answer
know. Were
that.
you snatched up by the pod people today?
Are you leaving us?
Well,
You
do you
want?
want my cells to dry out from
No,
plaque
no.
assay?
Go take care of
Otherwise, I
yourselves.
have to go. They need to have overlay. Otherwise, they'll dry out. And
Okay.
these are important. These are important experiments because,
Okay, go for it, Amy.
yeah, I forgot why. They're
Good
important
night.
experiments.
Good
All right,
night.
I'll see you tomorrow.
Yep. Bye.
Bye.
We don't want to show that yet. It's not over. Jamie says I'd call in sick tomorrow. Yeah, I understand what you're saying. Yep. Thoughts on the millions of shots that are being thrown away because no one's taking them? I think it's very sad, right? We have a surfeit of vaccines in this country. We should give them away. Nobody wants them. If 40% of the U.S. doesn't want them, give them away to a country that needs them. I'll go and give them if you want me to. How long after receiving a booster are you immune? So the maximum immunity is going to happen in a couple of months as your immune response matures. But within 2 weeks, you should really see the effect of that booster. Yeah. Masking in the subway just seems like a good idea. When they tell me I don't have to, I won't. But maybe some people still will. It's an interesting thought. There's a lot of weirdness on the subway, that's for sure. And a lot of smells. Boy. Okay. Does COVID vaccine and or virus cause herpes to activate? Okay, so herpes will be activated by stress. And what kind of stress varies. It can be sunlight at high altitude. You go skiing, you get UV light, you get reactivation. You get a fever, you have an exam the next day. All that can cause herpes reactivation. And so, yeah, COVID can do it, and so can the vaccine, because the vaccine causes inflammation, which is an immune reaction to the vaccine, and it could reactivate. Yeah, absolutely right. I've heard this not only for herpes simplex, but for shingles as well, for sure. When will we see more specific T-cell tests like we see with antibody testing? For example, T-cells specific to a pathogen to confirm responses. Yeah, for people with immunodeficiencies, leukemias. Yeah. Well, we have one T-cell test for SARS-CoV-2, right? Which just returns you a plus or minus answer. And I think that even can be improved. I think... So currently the way you measure T-cells is very laborious and doesn't suit itself well to a at-home format, right? So we have to develop better ways. I'm sure it can be done. People are very clever when they have to be, and when there's money involved, you know. So I don't know when, but I do think COVID has given a boost to these sorts of rapid assays, and it could be within the next few years. Any opinion as to why mortality from COVID is the highest in the world? Yes. Well, I'm sure you mean mortality. I think we have a lot of cases, right? Who's got the most cases in the world? Total cases. Let's look it up. We can look at the Hopkins site and see who. So, 28-day Germany is currently up there, right? So we have 79 million total cases. Yeah, that's the top. And we have almost 1 million deaths in the U.S., which is also in terms of raw numbers, right? It's the highest. So why is that? Why do we have I think, well, the sheer numbers is one, right? And you have so many cases, you're going to have a lot of deaths. But I think we also have a lot of elderly people, a good fraction of the population where the death is higher. You're more likely to die if you're over a certain age. I think we were slow to get moving on preventing and testing. And now a lot of people are not vaccinating. So every time we have a wave, we have more deaths, even from Omicron. Omicron can kill you. It does. I don't know why we say it's mild. So anyway, I think that's all of those things together. But I also think that there's underreporting in a lot of countries. It probably doesn't come up to the U.S. numbers, but there certainly is underreporting. That's what I think. Okay, Rob is a fungal lover, a fungus lover. And he wants to know what kind of viruses attack fungi. So mostly they're called mycoviruses, and they mostly are within the cells. They don't leave the cell. They're typically RNA viruses, and they reproduce intracellularly. And when the fungi divide, they divide with them. And they don't leave. They don't form virus particles. So they don't infect. But there are a number of those. And they can damage the fungus, or they can make it. So here's an example: chestnut blight fungus. A fungus that kills chestnut trees. We used to have a lot of chestnut trees in the U.S. Most of them were killed by chestnut blight. And a fungus, this has nothing to do with viruses, but actually it does actually. A virus infected them. It's called a hypovirus because it reduces the virulence of the fungus for the chestnut blight tree. So the virus reduces the ability of the fungus to kill the tree. Anyway, so that's one of the effects that they can have on fungi. Dr. Bailey says on her channel, scientists have no evidence viruses actually exist. So have I worked for 40 years on things that don't exist? I guess it's possible, but I know a lot of virologists, and we can't be fooling everyone. The NIH spends a lot of money on virus research every year. Viruses make people sick. We can see what they look like. We can grow them in the lab. We can see them in a microscope. We can sequence their genomes. They're distinct from cells. So what are these things that are infecting every living thing on the planet? Every organism has some kind of a virus. So exactly what are these if we have no evidence? Of course we have evidence. We've had evidence since the late 1800s. It's almost not worth answering. It's such an absurdity to say that they don't exist. They don't exist for you, perhaps, because you haven't read a shred of information about them. Or maybe you're just trying to be contrary, like a troll. Oh, what a concept. What a concept. Anyway, they do certainly exist. And I have a passion for them that is amazing, and they drive me. That's why I'm here. I wouldn't do this if they didn't exist. How quickly will new monoclonals be produced? You can make them very quickly. It just takes a few months. You can isolate B cells from people who have recovered from infection, say, or been vaccinated. You could pull out the antibody coding genes, make the antibodies in cells, and then test them. It doesn't really take that long. Yep. Now, genomic toxicity from COVID vaccines. Now, if Amy were here, she would say, I don't know what you're talking about. What shots? But I know exactly what you're talking about. There's no genomic toxicity from vaccines. The latest paper out of Sweden, which says that the mRNA can integrate, is nonsense. They use a cell line that has a lot of reverse transcriptase in it, which is not the case for terminally differentiated cells like your muscle cells where the vaccine is going. And anyway, your cells are full of messenger RNA. And that periodically can be reverse transcribed and integrated into the genome. Yeah. And you know what? Sometimes that results in a disease where the DNA integrates in the wrong place, but it's exceedingly rare. And just think, given all the mRNA we have in our cell, how rare it is. So I don't think this is an issue with mRNA vaccines at all. I don't get the insistence on masking in the subway. No one talks to each other. Well, not just talking that makes aerosols. Breathing makes aerosols, Jamie. But you're probably making a joke, so that's fine. I get it. I think also a lot of Americans have, obesity, and that's part of the high mortality. Could be, yeah, could be. Cases are going up in the UK again. So here's the thing, Mark. So there are going to be a lot of infections, always, because vaccines don't prevent infections. And most of them are going to be mild. And the ones that are serious are going to be either in unvaccinated people or people who are immunosuppressed or elderly, right? But as long as we test, we're going to see ups and downs. If we tested for rhinoviruses, we'd see ups and downs every year, every winter. And, we sort of see that with flu because we do moderate testing for flu, but not as much as we're doing now for COVID. So I don't think that's a concern, that it's going up or down. All right, to clarify, I know flu cases remaining low until COVID-2 wave subsided was attributed to human behavior. But here in the southern U.S., nobody was masking even during Omicron, and there was no flu. That is interesting. Nobody was masking. Are you sure, German opera singer? Show me the data. I think that's an anecdotal data point. It's not a data point even. How do you know nobody was masking? You went out one day and you saw nobody with a mask. But how about in the greater population? You'd really need to quantify that. And so I don't want to make a conclusion based on you saying nobody was masking. That's not how science works. It's not how we make conclusions. We collect data in a very careful way. So that's that. I live in upstate New York. I'm looking at the CDC criteria for masks. I am annoyed mostly that % hospital bed use is a factor. It makes no sense in places with the most hospitals across. Yeah, I agree. And Daniel didn't like that either. He didn't like the changes in the criteria for masking at the CDC, and also that they don't use testing, % positivity in testing as an indicator. That's not good either. Because if you don't test, then you're looking good. Why is no one testing anymore? I think it's because nobody cares if they get mild COVID. And if your employer doesn't say you have to have a weekly test, you're not going to do it. I think if you get symptoms, then many people will test. But if you don't, you're not going to just test randomly because you're going to be positive at some point. As I said, the vaccines don't prevent infection. And so I think that's part of it. Should we be concerned that B2 is 20% in New England, 10% in New York? Look, if it were 100%, I wouldn't be concerned. It's just another variant taking over, right? It's another variant that's more fit than the last one. And as we have said for months, for 2 years here on this live stream, being more fit doesn't have many necessarily implications for human health. It's just one virus is outcompeting the other. So no, you shouldn't be concerned about that at all. So Jack now presents WebMD data, 5.6 days versus 3. So it's not a progressively shortening incubation period, as you said. And the onset of, I think these are very difficult numbers to get. You really depend on people recalling when their symptom onset was, and that's very inaccurate. However, Daniel has mentioned this, that he thinks the time to onset might be around 3 days. And you said, would that be a form of evasion? And so, yes, in a way, because a rapid symptom onset would be a way of, so virus is presumably replicating more quickly, although we don't know that. There's no evidence for that. And then it beats your memory immune response, right? So that could be a mechanism of evasion. Just like antibody evasion is a mechanism of having a higher secondary infection rate, say in a family. But I don't think that's a pattern. I think that your original question, is this something we expect to continue? No, I don't think at all. I think you could make the argument that it would be an effective way for a virus to have a higher secondary attack rate, for it to reach symptomatic levels before immunity kicked in. But if you're asymptomatically shedding, that's all you need. There's no advantage to this, in my view, right? Correct. Florida is recommending not vaccinating kids, despite the fact that 800 kids have died in the U.S., and many of them recently. I don't understand how they can do that. What if your kid died of COVID and you had listened to the Surgeon General of Florida? These are not individuals who are speaking as responsible physicians or scientists. They know that it will get them votes, bottom line. There's no science that says you should not vaccinate kids. Thank you, Pamela, for your contribution. I am glad we are like a virology lullaby. That's great. That's just great. Do masks work? Yes, they are not 100%. No mask is 100%, and masks are different. So they may be at best, an N95 will be 80% efficient at preventing infection, especially if it's fitted properly. But if it's not, remember, and a mask blocks the droplets that you are encountering that carry virus, not the virus particles themselves. So they do work. You will always have fewer infections with masks than without. Studies show that, but it's never 100%. Can people who've received varicella vaccine one day develop shingles? If it's not sterilizing. So chickenpox vaccine, right? It's a good question. I don't think we're enough years away from the deployment of the chickenpox vaccine to know that, because it takes 40 years to see substantial shingles. It's a good question. Nobody has an answer yet, but at some point we will. Yeah, I think you're listening. The non-sterilizing will let a little virus in and establish latency. Right. Good point. That's a good question. I like that. Got to remember it. It's going to be a good virology exam question, right? There's a myth going around that 80% of people who died from COVID were obese. 43% are obese, 29% overweight. People who died from COVID, 46% were obese, 27% overweight. Well, you can get those numbers. You don't need a myth. You just can look at the numbers. You have the numbers right there. So you can. There's no need to go with myth for sure. Daniel's daily COVID death rates in New York seem to be 5-fold higher than Governor Hochul's. Can we get to the bottom of this? Just ask Daniel. I would believe Daniel. He has access to very good data. I got COVID 6 weeks ago, vaccinated, boosted, have high blood pressure, only got tiredness and body aches. Good. You didn't get long COVID either. That's good. Vaccines work. By the way, I was at Hunter College last week. I was teaching in a class, and my host is a neurobiologist, and he was talking about how infection could cause loss of smell and taste. He said if you don't recover, your sense of smell, taste within 3 months is gone forever. It's not coming back because the neural connections are hosed. They're not going to come back, which I didn't know. It's a tough one, right? how is it that the New York State COVID tracker shows 100 million persons? Maybe it's person tests, right? So one person could be tested 5 times, not just unique people. That's what I would guess. What's your speculation on why cases in the UK never really subside? They're not going to, because vaccines don't prevent infection. So as long as you're testing people, only test people if they have symptoms and you feel they're at risk, and you need to know if they have COVID and therefore treat them with monoclonals or antivirals. And so they're probably testing a lot. I know Wales has one of the best testing programs in the world. I had, what's her name? Catherine Moore. She's the head of COVID testing in Wales. I had her on Twiv a while ago. Very impressive what they did there. So they're really good at testing, and I think that's part of it. Most countries are not good at testing. How many shots do I need to be safe? Two plus infection, 2 plus a booster. If you haven't gotten COVID and you've traveled and you feel you want to play Russian roulette, then go for it. But if you are traveling a lot, I'd get vaccinated. Yes, I heard today, who told me? Yeah, Daniel said Ukraine had only a 30% vaccination rate. And they are, yes, the migration is causing a big outbreak, for sure, you know, the people leaving the country. And apparently there was a big anti-vaccine campaign run in the Ukraine, not by Ukrainians. So you can maybe figure out who did that. Spacing between first and second Shingrix for individual with comorbs. I don't know. I think that's a Daniel Griffin question. So I'm not sure about that. And, you know, as areas, many parts of the U.S. never masked very much. And listen, in the end, if you are a masker, what you are concerned about is either yourself, if you have a reason to be susceptible, or people who are near to you, young kids who can't be vaccinated, or older people, or people who are immunosuppressed, and that's fine. For everyone else, the virus will get them at some point. It will infect them, and hopefully they'll survive. Any comments with respect to nuts and bolts of testing and antiviral treatment? The test and treat, right? I think this is a mistake, and that's why David Ho said he's afraid of resistance. I said months ago that there would be resistance to these antivirals if you use them too much, and I don't think the test and treat is a good idea. I don't think anyone should have access to Paxlovid, because I think it should be reserved for people who are at risk. And now you may ask me, how do you know if you're at risk? Well, there are obvious indicators, right? And we know them all here on this live stream. But then you say, well, what if you are, but you don't know it? Yeah, well, that's a good question. And I don't have a good answer for that. But I don't think test and treat is a good idea. Daniel loves it. I think his opinion, yeah, he thinks the more people treated, the better. What changes the global pandemic situation? Just immunity? I do think so. I think immunity and vaccination. There are a lot of countries that haven't had access to vaccine, and that will change, but it will take a while. So, yes, eventually enough people will be immune from infection or vaccination, and it will not stop infection. It's never going to stop infection. It will stop severe disease, right? But infection is always going to occur forever, and mostly it'll be mild. White-tailed deer in Ontario, new variant or... No, it's a... So this is a preprint which I looked at this weekend, or last week. They had a variant in deer, which had a specific genomic signature, and some evidence that it went into one person, but it's not great evidence. One person. So I'm not worried about it going back, really. Amy, I recall 5 years being mentioned. I think by Stephen Morse. NIH said 5 years too. Yeah, so today I asked Steve Morse to come back because he apparently in 2 weeks he's giving a talk at Walter Reed about what we got wrong in the pandemic. I want him to come on TWIV and talk about that from him because I really like his perspective. And yes, I do think I wish Paxlovid could be used in combination because combination therapy greatly minimizes resistance, but we don't have that right now. No. Well, people are talking about stress and hair loss. I just started losing my hair when I got my job at Columbia in 1982, and I used to lecture, and my colleague, Saul Silverstein, said every year when you turned and write on the board, back then we wrote on the board, he said your bald spot got bigger and bigger. I said, gee, thanks. So was that related to the stress of a new job? No, I don't think so, because I loved having a lab to myself and doing experiments. I mean, there would be a 10-minute period where I got kind of stressed, but it's never a chronic thing. I'm lucky, I guess, in that sense. But you know, from the front, it looks like I have a full head of hair, right? But yeah, in the back, there's a bald spot. Why are people who beat cancer years ago and are not on immunosuppressants more vulnerable to COVID? The immune system has presumably recovered. Well, maybe there was some damage, right? It depends on what kind of cancer it was. If it was a cancer of immune cells, it may have done some permanent damage. So it's hard to guess, right? I'm not sure we understand that. I wouldn't be worried about the variants in the white-tailed deer, but I do think we should keep an eye on them. And what should we look for? Any changes that look like they might enhance reproduction in humans, because we know what kind of changes would do that. And it would be best if we could take them and put them in virus and infect cells in culture in the lab and see what they do, human cells in culture, right? Happy to report I was wrong and Vincent was right. After mask mandate ended, still so far significant masking in my sub-average vaccine uptake area. Very good. And I think that's Wisconsin, right? Are there any studies showing vaccine mRNA still found in cells months after vaccination? No. They looked very carefully before the vaccines even went into people, and they disappear within a few days. So no, there's no month-long persistence of vaccine mRNA. You know, mRNAs don't last forever. They all get degraded at some point, and the speed of that depends on the sequence of the mRNA. So this one is not designed to last a long time. Are there valid concerns about Chinese vaccine? What kind of concerns? What are you worried about? The numbers, we have some numbers, they're reasonable. They're not in the 90s in protection against severe disease, but they're good. They're better than nothing, right? It's what they have. I think it's fine. I don't have any concerns that I'm aware of. I haven't seen anything that would make me concerned. No. And are there viruses that can cause hair to grow back? The virus that calms you down, that removes your stress, right? That one would make your hair grow back kind of indirectly. And in both cases, I don't know. Speaking of raccoon dogs, commonly sold in wet markets. So tomorrow on Twiv, and Amy is joining us tomorrow on Twiv. It will be me and Dixon and Amy. And we are going to do the paper where they surveyed wildlife in China for viruses. And raccoon dogs was one of them, if I remember. Yeah, I like arancini. Yep. But here, as you know, it's not good. Any evidence of AFib being a comorbidity? It's a good question. I don't remember. Let's look it up. AFib and COVID. I just... Afibrillation. No, they don't like AFib. I remember Daniel saying this. COVID-associated atrial fibrillation. Yeah, here's a paper. It's a correlation, right? So here's one in 2021, December. Outcomes of AFib in patients with COVID pneumonia. So yes, there is evidence. David. Why do we need to get vaccinated against some viruses like polio only a few times during childhood, but in the case of SARS-CoV-2, multiple doses are recommended? Well, first of all, we're not boosting every year for SARS-CoV-2. We've boosted once, and that is enough. And any country who wants to boost every year is wrong. It's the wrong measure. And for SARS-CoV-2, it's a 3-shot vaccination, and that's the same as polio, 3 doses. So, I mean, flu vaccine you get every year because it's a lousy vaccine. It doesn't last, it doesn't have memory, it doesn't endure. But this one is not much different from polio vaccine, 3 shots, right? 3 times, and you get SARS-CoV-2 3 times, and that's all. So I don't agree that you're getting multiple boosts. Not the way it's going to work. If we have another pandemic and we're all wearing masks, will we have another pandemic? Well, we're not going to be wearing masks, right? We already know that. Most of the U.S. is not wearing masks. But if we do a theoretical experiment and say if 90% of the U.S. were wearing masks, that would make a big impact. Yeah, absolutely. But never happened in this country. Are T cells doing anything? Why are antibodies so overrated? So T cells are most certainly doing something. And let me give you the example of Omicron. If you are just double vaccinated, your antibodies don't neutralize Omicron very well. Yet, when you get infected, you're protected against severe disease. I think that's because of the T cells, because the ability of the T cells to eliminate infected cells has not been changed in the variant by the changes in the spike protein. So I believe the T cells are doing that. Now also, the first time you were immunized in the first vaccine trials, had immunized, vaccinated people, and they had controls. And they looked at the rate of infections, and at about 11 days, the rates diverged. And the rates of infections in the unvaccinated people went up, whereas the ones in the vaccinated people remained low. And that's before you could detect neutralizing antibodies. So it's either non-neutralizing antibodies or T cells that's doing that. So yeah, I think T cells are doing a lot of these. And why are antibodies overrated? Because they're easy to assay. It's really easy to do a neutralization assay for antibodies. It's not easy to do an assay for functional T cells. Is it true that Pfizer or one of the developers wanted some data kept private for 75 years? I have no idea. I really don't know what's going on here. And it's going to be hard to get into the root of that with Pfizer, right? Nobody's going to tell me anything. So I don't know what's going on. All I know is, as Amy said earlier, the vaccines, they work, not only work, but they're exceedingly safe. And if there were an issue, the FDA would have pulled the plug, but they didn't. So there's really no issue. And I'm not sure what the importance of this is. Companies always keep things secret. Besides Amy, who's doing important virus research currently? A lot of people are. I don't want to name names. A lot of people are doing really good virus research. I'll tell you what, whenever we do a paper on TWIV, I think that's good research, because I have a choice of a lot of papers. So if you hear us talk about it on TWIV, it's good research. Can you help me argue the point that it did not come from a lab leak or was manufactured in one? Nobody had SARS-CoV-2 before December 2019. It was a complete surprise to all virologists in the world. No one had it in their lab, so it couldn't have leaked from a lab. None of the samples in the Wuhan lab had SARS-CoV-2 in it. Now, you could say they didn't tell you, but that's not evidence. Give me some evidence. We have evidence that the virus is exceedingly close to viruses that are found in bats throughout Southeast Asia. Not in labs, they're in bats in the wild. And that's evidence that it came from bats and probably at a wild meat market. Next week, we're going to have Michael Warby on to tell us why he thinks the Huanan marketplace was the epicenter of the outbreak. It started there. Was it made in a lab? No. I have been making viruses for 40 years. I was one of the first people to develop the tech to engineer a virus, poliovirus. I did it first, making an infectious DNA of poliovirus. It all started with me 40 years ago. I stand on the shoulders of giants, though. I must have to admit, I didn't do it all on my own. Anyway, you can't, you change a virus, you mess it up, because you have no idea what you're doing. So to make this, they would have had something really close to SARS-CoV-2 to begin with and did something to make it virulent for people. There's no way anybody knows how to do that, and there's no close virus for them to do it with. No way, and I'm telling you as a virologist, it's just impossible that this was manufactured. So it came from nature, like every other virus on the planet. Human virus came from nature eventually. There's no evidence and no logic to saying it came from a lab or came from nature. Did you hear that researchers found the Endurance in the Weddell Sea, the ship that sunk 100 years ago? That's cool, like the Vasa, right? But that was even older, that sunk thousands of years ago, and they pulled it up. They pulled it up. When I went to Sweden, I saw it. It was just amazing. Really amazing. I found threading the needle interesting, informative. Have you considered another TWIV field trip? We've done a lot of field trips, but we can't, in general, get into BSL-4 labs because once they're operational, we can't go in. And the needle was not operational at the time. That's what we got in. But Rich Condit and I went to Galveston, where they have BSL-4s, and they put suits on us, and we've published pictures of them, and we did a TWIV about it. Yeah, we did a lot of field trips up until the end of 2019, and even a couple in the beginning of 2019, but we haven't done anything since then. Hopefully we start. I have a field trip planned for Madison in July. We're going to do a TWIV at the meeting of the American Society for Virology there. And so presumably we will do more of those in the future years. Yep. If viruses exist as a quasi-species in a host, what value do virus isolates have in terms of comparing one variant isolate to another? Okay, so yes, you're correct that viruses exist as a quasi-species in a host. In other words, when I'm infected with a virus, every virus particle has a slightly different genome sequence. But many of those don't make it. Many of the mutations are lethal. Many are neutral. Doesn't matter. So, yeah, the variant that comes out of me is going to be slightly different from a variant from you. And what we get as the sequence is the consensus, essentially, of all the different viruses that are coming out of us. And that isn't ideal, but we at least know, for example, changes in the spike that may be beneficial for the virus in terms of binding or stability. We can see those, we can put them into new viruses and confirm their effect. So there is value in having virus isolates because you can see the changes that occur over time. It's absolutely essential. Yeah. It's a good question, though. So many of you are asking about the interview. So I think it went well. It's just a matter of her getting a formal offer, and then hopefully that happens soon, and she will then have her own lab at some point. Hopefully she can continue the streams and the TWIVs. I don't know if she will. We'll see. Would severe initial symptoms, severe sore throat, great fatigue, anorexia, justify antivirals and even a triple vax? It depends. That's a call by your doc, whether you have, how old you are, whether you have comorbidities or not. And you err on the safe side. I know young people who get antivirals. I encountered a young person last week who, in her 20s, she got COVID. Her doc gave her a prescription for Paxlovid, and she took it. And she said overnight her symptoms were gone. So she's not got any risk at all. So it's your call of your physician. But I would say if you're over a certain age, it's not a bad idea, right? Amy has a limiter on her mic. It should be fine. I don't think that's an issue. Is a 12-week interval too long between first and second vaccine for a 5-year-old? No, it's not. It's better for immunity. So that's 3 months. 6 would be optimal. You have to balance it with the potential of getting infected. But if you say the community is 90% vaccinated, if the infection rates are low, then the longer the better. Yeah. You think Broadway shows should stop testing everyone every day? Yeah. No longer needed. They should be all vaccinated, right? And they should not be they should not be testing every day. No, I don't think that's necessary. And someone says, don't argue with the lab leak supporters. I'm not arguing. I think every opportunity is important to take because who knows who's going to hear it. Okay, that's how I view it. And it's not an argument. I'm just talking. I'm not having an argument with anyone, right? Yeah, there's a drop in myo between doses 1 and 2. That's absolutely right. And the booster is much lower. Absolutely correct. Yep. Hello, Vanity. Good to see you again. Thanks for stopping in. So now we have all our mods except Frank. Good to see you. Amy didn't get a new job yet, but she interviewed and it's looking good, which would be great for her. I would love to join the Q&As after COVID. Oh, you mean listen? Yeah, we're going to do other things. I mean, we'll talk about all viruses. Oh, here's the thing. Here's a thought, folks. I was thinking of a new live-streamed course on coronaviruses. Let me know if that's of interest. I'm going to do a viruses one in the fall, but if I don't teach at Columbia this summer, then I could do a live-stream coronaviruses course. So let me know if that's of interest. If you don't want it, that's no problem. is the virus on TWIV mug a polio virus? It sure is. That is a poliovirus with its receptor attached. So normally it wouldn't look like that, but it's the model. And I get to show you, that was the icon for TWIV since the start. But let me show you the model I had made of that. So I had a 3D printed model of the same thing, poliovirus, bound with its receptor. It even has a name on it. See, poliovirus plus poliovirus receptor. And this one, same colors. This one opens up. You can see the inside. Oh, wouldn't it be cool if I had the genome inside? Yeah, that would be cool. Anyway, you can see the thickness of the shell. This is going to the incubator at some point. But these are all receptors here in blue. And so I used this model years ago when I was looking for an icon to, make on TWIV. That's what I used, and I use it to this day. Well, the microbe TV icon is different, right? That is this. I had this made by an artist, and she thought, since I love plaque assays, you see, she listens. She knows I love plaque assays. She made the O a plaque assay. So that's microbe TV, but the TWIV is polio, as it should be, because it's the virus I worked on for years, right? Okay. We got over 500 people tonight. How cool was that? How cool is that? That still people are showing up. It is going down, but that's fine. Now, this is a tough question here. What are the odds of a triple vax 53-year-old woman with diabetes and CKD stage 5 surviving COVID? Really hard to know. Really hard to know, right? It's probably higher than if you were a healthy 53-year-old triple vax person, but I don't think anyone could give you a number. You should be careful, right? When Amy says she's against boostering, does she mean you don't need a booster if you have properly spaced vaccines? Well, no, I think she thinks even with the improperly spaced, you don't need a booster. Because as Paul Offit says, there's still no evidence that without a booster, you would not have good protection against severe disease, even with Omicron. Now, we both saw the results of a study earlier this year, which she said that the booster fixes the too short spacing in terms of antibodies, but we don't know the effect on severe disease. So that's why. And Paul Offit feels the same way, and she's with that. But, the day after we got our boosters, which was, a week after we announced it here on the live stream, then Columbia made it mandatory to have a booster, so we would have to get one anyway. By the Pfizer documents that anti-vax pundits are pushing are being taken out of context. Of course. You should really be careful. Don't listen to the anti-vaxxers, Malone and Mercola and Wakefield. Don't listen to them. They have an agenda, which is to get money, to be paid to say this stuff. And they don't care about your health. And so, you may say, I want my right to vaccinate myself. They don't care. You should make a decision to vaccinate yourself. By the way, a TWIV listener sent me a image. I got to see if I can find this. It's actually in the show notes for tomorrow's TWIV, so maybe I could find it and show it to you. Hold on. It'll just take a minute. TWIV papers 875. Here we go. This is just great. Oh, I don't even have to download it, do I? No, I don't have to download it. So I can just pop it in here. And look at that. How do you This is funny. Warning, bridge washed out. Unless you feel that this is an attempt to deprive you of your liberty, then let me move this out of the way, then keep driving. Now, of course, this is photoshopped. You can tell because the sign thing here is just too perfect, right? But it's still funny. I like it. Anyway. Yeah, one of the VAERS reported effects was turning into the Hulk. You can report anything you want on VAERS. And then the CDC goes through and tries to validate them. Most of them are thrown out because they're garbage. Are you optimistic about a possible universal flu vaccine? I am. I think we can have one in the next five years or so. Yeah. All right. 3 times vax. I have everything delivered. Stay out of public places. If I do have to go in, I wear a respirator. I have comorbids. Old parents rely on me. Should I relax? Although if you have old parents who rely on you, you can be careful for them. I think that's fine. I don't think you're being over cautious there. My adverse event was verified through VAERS. There you go. So it can be verified by contacting people. I do know they monitor us for over a year, went through doctor reports. Excellent. So the proper ones are monitored properly. They're doing their job. So let me tell you a Joy Division story. I also used to love them. When I was a postdoc in Boston, there was a postdoc down the hall. He and I used to love them. We used to go out and listen the radio, the music playing in bars and stuff. And then one night they came to one of those small venues in Boston. Someone right outside on Commonwealth Ave, right outside of the city. What was the name of it? I can't remember. It was tiny. It was in a basement. Anyway, we saw them. In this tiny room, 3 guys. They walked right next to us and went up to the stage and they played. Except by then the lead guy had committed suicide. So technically it was no longer Joy Division. It was what they became. I forgot the name, and I'm sure you guys are going to tell me. But I really liked them too. We saw what was left of them, their remnant, yeah. If Amy likes Joy Division, she will like the wonderful amethyst Kia cover of Love Will Tear Us Apart. Yeah, I haven't heard that. She will hear this now because she listens. There you go, Amy. Masking mandates will be gone in Ontario March 21st. I'm nervous. Fully vaccinated, boosted, but nervous. Don't be nervous. You'll be fine. You will be. If anything, you'll get a mild sniffle. No worries. Yeah, it's Transmission by Joy Division. That's right. There is a song called Transmission. Yep. I have the vinyl of their albums. What's the name of the group? New Order. Thank you. New Order. Yes. So, because I used to say I saw Joy Division, and I got into an argument. He said, "No, you didn't. If the lead singer was gone, you didn't see Joy Division." I said, okay. They didn't call themselves New Order at the time, or maybe they did. I don't remember. What was that club down on Commonwealth Avenue? Somebody's got to know this. It's not there anymore. It was a tiny thing in the basement. All right. How do viruses combine if they are not living and do not mate with each other? Well, 2 viruses walk into a bar. No. 2 viruses walk into a cell. They enter a cell, and their genomes begin to multiply. In the process of multiplication, the genomes get mixed up, and you get parts of one genome with the other combined, and then the new viruses have that combined genome. So they're not really mating, because mating has a specific meaning, right? It's usually sexual reproduction. We're talking about mating. So viruses don't do sex. And so that's how it works. You don't have to mate, and you don't have to be living to recombine. You just need to be able to reproduce your genomes. Okay, now we're talking about VAERS. Is there any worry about gain of viral mutations of SARS creating far more lethal variants? No. No worry at all. Why do I say that? And you should ask me, why do you say that, Vincent? Because first of all, it's never happened, which doesn't mean it won't happen, but there's that, and you always have to go on what's happened. That's what we do in science. And secondly, there's no selection for being more virulent. I cannot think of a reasonable selection for being more virulent. I can think of one for being less virulent, but not more. So what would make a virus more virulent? An accidental mutation that caused some other change that was good for the virus, maybe. Yeah, and that would have to be rare, I think. So I'm not worried about it. I haven't seen a more lethal variant arise yet in SARS-CoV-2. Nobody's proven that any of them are more lethal than others. I've been reading about the use of viruses in cancer treatment and gene therapy. Perhaps a benefit of the pandemic will be increased research findings. So before the pandemic, there was a good amount of research invested in oncolytic viruses, viruses that treat cancers. Why? Because they're promising. And so cancer, as you know, attracts a lot of money. So a lot of money has been invested in it. And I always say in the next 10 to 15 years, people will be treating many different cancers with viruses. So I don't think it needs to be increased, but if it does as a consequence, that would be great. Yep. Is Omicron less virulent, or does it just infect more people protected against severe disease by vaccines and previous infections? Yes, it does. Whenever you see, the South African initial study, yeah, it's less virulent because most of the people in that study were vaccinated. Now, if you're not vaccinated, it is just as virulent as all the other SARS-CoV-2s. As Daniel says, it can kill you. It can kill unvaccinated people. It kills kids. More kids have been killed by Omicron than before Omicron emerged. Okay? Think about that. Omicron kills. It's not less virulent. It's not less virulent. Should I say it again? Interesting. People who don't trust vaccines in the government are people who have been hurt by authority figures and do not trust authority. Okay, I'm not an authority figure. Vaccines are good for you. I'm not an authority figure. I have zero power in the greater scheme of things. Zero. Well, maybe 0.1 power because you guys are here and you want to learn. I can teach you. I have some power to teach, but I have no authority, so I say vaccines are good, but they won't listen to me either because they say I have expertise and they don't trust expertise either. So I don't know what to do. Don't know what to do. I got a booster a long time ago because as I just mentioned why, so I don't want to go through it again, but then the next day Columbia said you needed a booster. Not sure it was needed, but I thought it was the right thing to do. And it's the last SARS-CoV-2 shot I will get. No more needed. I'm good. I'm going to get boosted every year by SARS-CoV-2, and so are all of you. You're going to get a mild infection, and that's going to be your yearly boost. Oh boy, now we're talking about someone else here, which has a large audience and makes videos. Good for him. In the future, will we have a new set of behaviors for dealing with colds and flus? No. People don't want this. Some people will have gotten used to wearing masks. That's true. But most people will not. We're going to go back to the old stuff. But maybe we're going to have better vaccines as a consequence, even against influenza virus, right? And that would be good. When I referred to Sam Bailey saying viruses don't exist, I wasn't trolling you. I merely pointed out that there were dangerous people out there. No, I wasn't blaming you. I was saying these people are trolling, the ones who say viruses don't exist. Hey, look, this is why I need your help to build Microbe TV so we can get more people listening, because I'm doing all the content myself and it's too long and it's too hard. I need people who can come and work with me and make short stuff that people will listen to that I say can be released because it's correct, right? You get that? That's why I try to raise money to hire people, and we're getting there. We're doing a really good job with your help, and we just need to keep going. I'm having a very good success with what we're doing, but we need to get other people involved so that we can reach these kinds of people who say viruses don't exist. We have to have more views somehow so that people hear us who say, yeah, here's a virus and here's how it works. That is, that's my goal, to be Earth's virology professor and then have all these people working with me who are Earth's other things as well. And instead of going to someone who doesn't say anything, they'll come to us. So that's why I'm doing this, and I need your help to do it. You should come here to learn about viruses like Mark does. This is where you learn. Everywhere else on YouTube doesn't know about viruses like we do because we've worked on them for so many years. Are they working on a COVID nasal vaccine? Yes, we did a paper recently on TWIV where had an interesting virus vaccine intranasally delivered that contains 3 different viral proteins. It was very good. So I don't know, though, by the time they get it developed, maybe we won't need it anymore, because I'm saying that this virus, this is going to become mild once everyone's immune to some extent, and not going to be many serious infections. Isn't discussion of variant mildness a poor issue, as the degree to which the virus infects or is less important than the inflammatory overreaction? Yes. So the inflammatory overreaction is a host thing. You're absolutely right. And that's where most of the severe COVID comes from. And so, yes, I agree with you. The problem is, could we can't test virulence in people. We can't take, well, we could. They did that in the UK. They could take young, healthy people and give them different variants and see if there's a difference. Maybe they're going to do that someday, but I wouldn't advocate it. I think that's unethical. So yes, I agree. It's a difficult problem, and this is one reason, but I also have many other reasons why it's just improper to say this variant is more virulent than the other, because you're always talking about different populations, as you say, and it's the host. That's the main thing. It's a scientific proof that masks don't work if you don't wear them. Very good. I like that. Varicella vaccine in children, shingles occurs significantly often. I don't know how you can tell that, because the vaccine is not old enough to be associated with shingles. I need to see that study or studies. How long does a virus last floating out in the open environment in the droplet before it's operable? In the droplet, well, the droplet falls to the ground relatively quickly, so it's okay until it falls to the ground, and then if it's on the ground and there's sun shining on it, it's not going to last more than a few hours. It really depends on the environmental conditions. If it's cold, it could last longer, but who's going to be sniffing off the ground? That doesn't really matter anymore. It's what's in the air that matters. Now, if you keep the droplets suspended by circulating air for a while, I think not more than a few days of infectivity in droplets. How much virus does it take to cause an infection? No, we don't know. The study in the UK, the challenge study, they said 5 PFU will infect half the people, but they had totally unrealistic infection conditions. They had pipetted the virus into people's nose, and they clamped them with a clothespin for a couple of minutes, which is not the way you get infected. So I think we don't know. No, we don't know. Do you think it was a public policy mistake to mandate masking that included cloth? No, I frankly don't think the difference between cloth and surgical is all that much. The numbers are very close in the studies have been done. I've worn a cloth mask the whole time, and only recently did I have to start wearing a surgical for Columbia because they required it. But I don't think the data say there's a statistically significant difference between the efficacy of the two. Is an organ transplant success the same relationship as a virus success? No, it's totally different. Organ transplant success is all about suppressing your immune response so the organ is not rejected, whereas a virus doesn't have to do that, no. Okay, we are reaching 2 hours. Let's start to wrap up here. We're close to the end. Is it 12-week interval? I already answered that question. Yes, sorry. Let's see. You love to listen to me talk. Well, thank you. I appreciate it. Not everyone says that. Matri, thank you for your contribution. Really appreciate it. What happened to Zika? Well, it's still around. It won't have dangerous variants. That's not how viruses work. They just reemerge if there's a lot of population immunity. There are outbreaks of Zika in countries where the mosquito is present, but we don't hear about it because the scare is gone, really. Wouldn't it be great if TWIV fans could have access to all the research papers discussed? Yeah, I'm sorry about the paywalls. You could always email the authors, they'd be happy to send you a PDF. They always are happy to send you PDFs of their work. Yeah. Do you think chronic wasting disease is going to kill us all? No, not at all. It's very rare. I mean, it's increasing in deer, obviously in cervids, but there's little evidence that it's going to spill over into people, so I'm not worried about it. And there are 6 deer in your yard. I have tons of deer in my yard, and I'm not worried about it. No. I look like a dignified senior virologist. I've often wondered if male pattern baldness has some weird evolutionary advantage. No, I don't think so. There's not enough time to have any advantage. And Vanity reminds us, if you want to donate, you can buy stuff. Here, you go to patreon.com /microbetv and you can donate, or you can go to cafepress.com/twiv and buy stuff like this mug. It's just water. Thank you, Ian, for your contribution. Someday I'd like to visit the incubator. Yeah, anytime. Incubator is shaping up. It's a lot of fun, and you are welcome to visit. And thank you for the Bluetti. It's a lovely piece of machinery. It's a battery, and I have to figure out how to get it to where it needs to go. But thank you so much for it. Really appreciate that. 1968 Mary Tyler Moore movie about a happiness virus? Really? I didn't know that. That's really funny. So we will never reach 1 in 100,000. No, we will. At some periods of time, right? We will. It was not going to be always, because in the winter, I think the case numbers will go up because this will be a winter virus in the Northeast in temperate climates. Yeah. So we will at some point, maybe in the summer, but other times we won't. No. Thank you, Doreen, for your contribution. Illinois. Oh, you're going to drive to Wisconsin in July. Well, thank you. Drive safely. A lot of good questions here. Sorry I can't get to them. I don't want to keep our mods here for more than 2 hours. And I don't want to keep all of you here because you guys are good. You stick around until I leave. I asked lab people about MERS, Western equine encephalitis, Zika, all the other emergents. Why aren't they saying the same about them? They've never heard of them. They're saying the same about this because someone started it in the press. and they think it's a good idea, but they have no idea. If you receive an antiviral and it clears symptoms in a day, does that reduce the infection immune response? Probably, yeah. Depends on how soon you got it after you were exposed, right? Amy deserves a good gig. She's a gem. Absolutely right. And hopefully she will get this gig, and she will be the master of her realm, and I think perhaps have less stress, although don't count on it. Now, Pete doesn't want a course on SARS-CoV-2, but a single lecture. Yeah. It's okay. I thought of a single lecture, but I want to do a course on all the coronas. I think that would be fun. And I don't have to stream it. I could just do it as a series of lectures. And Charles, yes, I know it wasn't you, but I was just saying, I don't think anything is bothersome. If the same question is asked over and over, I'm happy to do it, because I want to get. I don't think there's so many people who can provide information that's useful about viruses, right? So, okay, most people would like to see a coronavirus live stream. So the live stream courses are cool, right? Because it's a lecture, but it's also you people can ask a lot of questions. Plus, you can chat amongst yourselves. Very unusual. No. So Pat, don't get the wrong impression, okay? Poliovirus does not look like this. Oh, you said I thought them were spikes. No, they're not. These are the receptors. But normally just the magenta would be poliovirus, okay? It's just that we did the structure of the thing bound to receptors. And I know it looks like a coronavirus, but it's not. It's not. Oh, what's the yellow virus thingy I see behind you at the incubator with the stringy things hanging from it? So we have two scientific terms there, thingy and things. That's poliovirus with its genome coming out. And the genome is beaded. So a virologist, Ann Palmenberg at Wisconsin, took the genome sequence of poliovirus and took 4 beads of 4 different colors and strung them on a metal wire in the right sequence. So that's the genome spilling out of the virus particle. I've often shown it up close. I can do it again so you can see it. It's really cool. It's very cool. I'd love to see a course in historical milestones. That could be another one, yeah. You know, I could take this. I could take this model and I could put that beaded genome inside. It would almost. So the genome of polio really fills the capsid, okay? And so that I'm not sure that beaded genome would, but that's a good idea to put it inside them. Yeah. Oh dear. Do viruses originate from animals, or do we breed them in slaughterhouses? No, we don't breed them in slaughterhouses, though. Pigs are good hosts of influenza viruses. So because the world pig population has risen so much, there's more spillover of influenza viruses into those animals. But it's not in the slaughterhouse. It's actually where they're just raised, for 6 or 9 months or whatever it is. An entire course on just polio. Yeah, that would be fun, wouldn't it? And I could have Neil Young. Yeah, I have to get more famous before he'll do it, though. He doesn't even answer. He wouldn't answer my correspondences. I had years ago. I had an idea for a podcast which would be seasonal, and every season there would be an infectious disease where we went through the whole history in like 20 episodes. And I still want to do that, but got kind of interrupted by COVID. But hopefully we get back. Thank you, 844OK, for your contribution. Really appreciate your support of what we are trying to do here. And environmental, thank you for your contribution. Really appreciate it. Let's be honest, the government lies and so do experts. Not this one. I don't lie. Not about viruses. How could I lie about viruses? This is my passion. I would never lie about them. I could be wrong sometimes, but not on purpose, because maybe I just am mistakenly recalling some factoid, but I would never lie about viruses. Vinny would never lie about viruses. Any progress on the Discord server? No. It's on my list. And if you know me, I'll get to it. Don't get impatient, please. Non-viable virus parts are everywhere. Yes. Would someone's immune system, if never introduced, identify and react? Depends on how many bits and pieces. I think you need a lot of bits and pieces to get the immune system awake. So it's not going to happen just randomly. Now, it's not the paradise. It's something else. And my friend who went with me, he died, unfortunately. So I can't even ask him. I'll figure it out. Micro TV could do short, sharp topics. Yeah, I know. Short, sharp. I need help to do that. I can't do all of it myself. I'm good at these long form things and teaching. The short stuff, I need help, but I'm sure I can get help. That's why we're raising money. Stay frosty. Well, thank you. I'm glad we're helpful. I try and be useful. Oh, yes, this is a good one. A virus goes into a bar. The bartender says, we don't serve viruses here. The virus replaces the bartender and says, now we do. Yeah, I've heard that one. That's a good one. Someone sent me an email. They said their kid has a bacteria joke. Do you know why bacteria are so good at math? Because they multiply really fast. You could say the same. You could say the same for viruses. Yeah, so Roger has a guy producing his videos. All right, I need a full-time person devoted to Microbe TV. When I make a pod, it gets produced, it gets released right away with no delay. And that's not going to happen right now. I need to have more substantial funding for that. Thank you, Graham, for your contribution. Difference between science fact and public health messaging. Oh, so I do science fact. I don't do public health messaging. And I get yelled at now and then because people say, oh, you're not taking public health into account. But I think that public health messaging needs to change to accommodate the fact. And if the facts are bad, you need to tell people. But if they're not bad, you shouldn't scare them. And I think the press does a great job about that. But that's a topic for a whole nother session, unfortunately. But it's a great point. Thank you, Kev, for your contribution. Really appreciate it. And thank you, Rob, for your contribution. All of you are helping the incubator to grow. The Mary Tyler Moore happiness virus. Yes, Ann Palmenberg beaded the coding sequence of polio virus for me, and she sent it to me a number of years ago. So as you visit the incubator, you'll see it. It's just brilliant, just brilliant. I wish Twiv would play the intro music longer. It used to. If you go to the pre-COVID episodes, it used to play longer. And people complained, there's too much time to get into the real stuff. Stop it. Oh, I'm telling you, you can't please everyone. So I made it short. In fact, there used to be a totally different intro, which I changed. And maybe this is a good way to end out. Do I need to get my 11-year-old son his first jab? Yes. He's negative from a COVID test. Doesn't matter. Get him his first and then his second jab. Absolutely. And Charles, that is kind of a backhanded compliment. I mean, I know you mean well. Of course I do it better than the Florida Surgeon General. Oh my gosh, the Florida Surgeon General is not interested in public health. You know what he's interested in. Hey, Steve, look at that. How are you doing? Did you get my email tip for suggested fungi twixter? I did. Thank you very much. It was on the list of TWIM today, but we didn't get to it. But we'll get to it next time. Are there any science communication charities? Yeah, there are some. We'll be approaching them. Yep. We just hit 400 likes. That's a good time to thank the mods for being here tonight. We have Les, we have Steph, we have Tom, and we have Vanity Nutrition. And thank you so much for moderating. Really appreciate it. Kind of a thankless task, but you do get to participate. And I want to thank everyone for coming. 536 of you, 400 likes, and thanks for all your contributions to the incubator. You're helping us to develop, the next era of science education. Let's put it that way. See you next week on Wednesday at 8 p.m. Meanwhile, have a great week and be safe. Thanks, everybody, and good night.